Bismillahirrahmanirrahim. Welcome again, guys. Welcome to another video lecture by Kami Biology. Today in this video lecture, we are going to talk about termination of DNA replication in eukaryotes. So let me write. Termination of DNA replication in eukaryotes. Termination is that stage where the replication is stopped and the two strands of the DNA which is synthesized are separated from each other, and we get two DNA strands. In two DNA strands, one is the parental and other is the newly synthesized strand. Similar in the other DNA molecule, we have two strands. One is the newly strand and other is the parental strand. That's why we can say that this DNA is usually replicated through a semi-conservative manner. So in the previous video lecture, if you guys remember, we already talked about that elongation of DNA replication in a detail. So let me tell you again that in what stage we are in the previous video. In the previous video, if you guys remember, we talked that this is the leading strand and this is the lagging strand. Now the leading strand are continuously synthesized because there's no issue, because DNA polymerase delta or epsilon can synthesize the strand in 5' to 3' direction. So there is no issue because this one strand should be synthesized in 5' to 3' direction. Okay, because this one strand is having the end of 3' to 5' end. That's why there is no issue. While at the lower end, if you guys remember, we talked that the problem are always created at the lower end. So at the lower end, we have a 5' to 3' end. So the new strand will be synthesized in 3' to 5' end. Okay, so there is an issue, and at this position there the primer is added. So many primer is added, which we call that Okazaki fragment, and from each primer there is a gap, and now this gap is filled by the DNA polymerase delta. So DNA polymerase delta can fill up the gap. Okay, and at the primer, if you guys remember, we talked that at the primer there is a hydroxyl group is also present, or 3' hydroxyl group is present, and they can add the nucleotide, not specially here at that specific point, sorry, here. Okay, and start adding many of the nucleotide we have. These are the RNA primers. So adding a nucleotide, DNA polymerase delta are bind here because there is the 3' hydroxyl end, so they can add the nucleotide in this direction and then leave it, and then hold another primer and add the nucleotide and then leave it. Okay, and we also talked that DNA polymerase delta have also a little bit endonuclease activity, so they can, when they reach, let's suppose the DNA polymerase enzyme reach at that specific point, let's suppose. So whenever they reach at that specific point, so then they have also the ability to remove some of the RNA nucleotides. So they are just removing some of the RNA nucleotide and we get a flap just like that. And after that, this flap is removed by a Fin enzyme, Fin protein, if you guys remember, we already talked about. So Fin can remove this one, flap. So this gap is filled. Again, we have some of the RNA nucleotides present. So again, the DNA polymerase hold this one side and add the nucleotide, and this process is carry on. Okay, and we also talked that these RNA primers should be removed. So the RNA primer should be removed by the an enzyme we call it RNase enzyme. So RNase enzyme can remove the RNA primer, as you can see. So the RNA primers are removed and the gaps is filled by DNA alpha. DNA polymerase alpha can can fill up the gap, and there is still the nick is present. So the nick is again sealed. Let's suppose if this is the 2 DNA strand and there is a gap is formed. So this gap is filled by, there is, let's suppose this is the RNA primer. So this RNA primer should be removed. Now the RNA primer is removed. This is the template strand, let's suppose. So this primer is removed. Now the gap is filled by DNA polymerase alpha. So DNA polymerase alpha can, they can fill up the gap by adding correct DNA nucleotide, but still the nick is present, as you can see. Now this nick is sealed by the DNA ligase enzyme, so they can seal the nick. means they can provide the phosphodiester bond. So when the phosphodiester bond is present between the nucleotides, so the DNA, the bond between the nucleotide are become stronger and DNA are become stable. This is how the process is carry on in case of the elongation. But remember, when this complex means activated complex, which we call it CMG, and it contain MCM, CDC45 and GINS. This complex is going on, but when they reach at that terminal side, not terminal, before the terminal side, when they break some of the nucleotide, then they contact some of the protein, and these protein are, they contact with some of the protein, and these protein are the SCF3 ubiquitin ligase. So, this complex can meet with this complex. This is also a protein complex. So this one complex meet with this one complex. So two complexes are formed. Now, what actually the function of this complex? These are actually the protein or enzymes. The function of each protein is that they can ubiquitin this MCM. So the MCM is now ubiquitinated. So let me draw the MCM here. So this is, let's suppose, the MCM. Now the MCM is ubiquitinated. Usually the 7 part, let's suppose this is the 2, 3, 4, 5, 6, 7. So the 7 part is ubiquitinated, means the lysine area is ubiquitinated. So here ubiquitination is happen. So protein are added here, which we call a polyubiquitination. So when the polyubiquitination is happen at that specific point, then all of you better know that this complex should be degraded by proteasome. But in this case, the complex is not degraded by the proteasome. Why? Now this ubiquitination complex is then meet with another protein, which we call that, so they contact with P97. This P97 is also called a VCP, villosine containing protein. Now this villosine containing protein is actually a segregase enzyme. Segregase enzyme. What are the function of this segregase enzyme? When this ubiquitination part of this complex are bind with this P97 protein, then they can disassemble this complex. So they can disassemble. So the structure of this complex is changed, or the structure is disrupted. Once the structure is disrupted, so the complex is degraded or removed. So the same complex is then removed out. So let me draw here once again the same structure. So we have again, this is the DNA strand, and this is, let's suppose, the terminal end. Okay, so this is the terminal end. So terminal end is also the terminal end is also separated from each other because the enzyme are reached at that specific point, so then it is removed. And again here the primer is added, means the upper strand is again synthesized. So this upper strand is continuously synthesized because there is no issue, but the lower end is again synthesized in a fragment form, which we called as Okazaki fragment. So at that specific position, at that position the strand is synthesized and the RNA primer is removed at that position. But at that position, when the helicase activity is done, or helicase activity is finished, then again the primer is added at somewhere at that position at terminal site. Okay, then let's suppose this is the strand is already synthesized. Okay, now again this gap is filled by DNA polymerase delta. They can fill up the gap. Now the gap is filled up. At some specific position at that specific area, as you can see, there is also the primer. Okay, and here we have also the primer. So remember, in the lagging strand, we have primers at the terminal end of the linear DNA molecule. Now this is the eukaryotic DNA. So in eukaryotic DNA, we have the linear DNA molecule, and in eukaryotic DNA termination, there is always a problem is usually created. Why? Because whenever the primer is removed, let's suppose this primer is removed, should be removed from that specific site, as you can see, both from the leading and from the lagging strand at the terminal side. Okay, so when at the terminal side the primers are removed out, then what actually happened? That specific area is also transcribed. But remember, here is what at that specific site there is the 5' end, and there is also the 5' end. This is the problem. Now the strand will be synthesized from 5' to 3' direction, and here the strand is synthesized in 5' to 3', means in this direction the strand will be synthesized, and at the upper strand will be synthesized in 5' to 3' continuously. But at the lower strand, the new strand will be synthesized in a discontinuous manner. Okay, but when the primer is removed, so how it is possible for the DNA polymerase delta that they can add the primer? Because here is the 5' end, not hydroxyl group, or not the 3' end. Here is the phosphate group. So how it is possible for the DNA polymerase delta that they can start the process of the elongation, or they can start adding the nucleotide? How is it possible for the terminal end? At that specific position, there is some kind of the sequences which we call the telomere sequences. So there are the sequences which we call the telomere. Try to remove the spelling mistake by yourself. Okay, telomere sequences. Now the telomere sequences are TT, G, G, G. Now these are the telomere sequences which is usually present at that specific point, as you can see. Okay, so it is present at that specific point. So let me write T, G, 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 and T, T, and G. Okay, and it is also present at that specific point as well. T, T, G, 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 T, 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 and G. Okay, and again here is also T, G, 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 T, T, and G. So at the terminal end of the DNA, there is the sequences which we call that telomere sequences, as you can see. Okay, now this telomere sequences is present, and there is this newly synthesized strand is a short, as you can see. So that area is also synthesized, means there is also some information is present. So these are the genomic area, these are the genes area, as you can see. So these areas should be also means at that specific area DNA should be also synthesized. So there the DNA will be also synthesized, but it is not possible. Why? Because here is the 5' end present. Then how this problem will be solved? This is a problem. So how this problem will be solved? So we have another enzyme which we call the telomerase enzyme. Telomerase. Okay. Now this telomerase enzyme have two activity. One activity is that they can add the nucleotide, polymerase activity. Okay. And the next activity is that they can catalytic activities also. So what actually happen? If let's suppose this is the parental strand, as you can see, okay, and this is the strand. So let's assume this one area. So I draw here in a large diagram. Then telomerase enzyme. Telomerase enzyme have a kind of sequences which is complementary to this telomere sequences. Okay, so they have a sequences like that. Let me draw. So its structure will be in this form. as you can see. So they have a adenine, cytosine, 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 adenine, 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 and cytosine. So this telomerase enzyme will be, bind at that specific point, as you can see. So there is also what there is the T means this enzyme are bound at that specific point. So we have the structure like that. So now it is bound at that specific position. So adenine, cytosine, okay, then cytosine, 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 adenine, adenine, and cytosine. And then one thing more, the nucleotide are bind here, okay, and they can also elongate the strand. So the parental strand is now elongated. So they have also polymerase activity. Now they have polymerase activity, they can also elongate the strand. So we have again the same situation here. So let me draw. Now there is also G, 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 and T, T, and G. Okay. So then it is again removed out and bind at that specific position. So it is removed, as you can see. Now it is removed. And then it is bind to that specific point. So again it can bind at that specific point, as you can see. Okay, and make a bond with the terminal end and again elongate the strand. So 2 times they can elongate the strand. Now what we have the strand is elongated. So this telomerase is then removed out because the strand is elongated. And here we have what? We have the newly synthesized strand, as you can see. Okay. Now this is the newly synthesized strand and they can make a pair. But remember, that one portion is the junk portion. Junk DNA. Here is no genes, here is no information stored. So that area will be not, replicated, or that area will be not synthesized. Means newly synthesized strands will be not present at that specific area. But still the primer is bind here. Primase can, add the primer here. So primase can add the primer and again we get the gap. Now here is also the hydroxyl group is present. Then DNA polymerase delta can comes and they can add the nucleotide. This is the DNA polymerase and they can add the nucleotide, as you can see. Okay. Same situation again happens. So they can remove the RNase enzyme comes and they remove this primer. So again the same situation happens. But remember, this is the flap. This is the flap, or this is the overhang site, our junction site of the DNA, and this site should be removed. Why? Because here is no information stored. We just need that area because at that area the information is stored. Okay. After that, fin enzyme. Fin 1 usually they comes and remove this one area. So this one area will be removed out. So we have just the DNA strand just like that. And that's how the terminal end is also synthesized. So that's how the terminal end is also synthesized. But not only the same process, not only the process is happening here, means not the telomerase is, performing the function here. The telomerase enzyme is also performing the same function here and here is also. So the same process is also, means the same process also happening at that terminal end as well as at that terminal end as well. Okay. And similarly, the termination of the transcription, termination of the replication is done. Okay. So that's all about the termination of DNA replication in eukaryotes. So I hope you get idea about the termination of the DNA replication. But before that, you guys need to watch initiation, elongation, and the protein which are involved in the DNA replication. Okay, then you will get idea about the termination of DNA replication as well. So it is present into my channel, or I will give the link of all the video which are regarded to the DNA replication. Then it will clarify all the concept about the DNA replication in eukaryotes. Now DNA replication, these 4 videos are very important for the MPhil, BS and PhD level student, because it is very complicated and complex process. And if someone is doing PhD, MS or BS means level degree, so they need to understand DNA replication in a detail, as I explained in my 4 videos. This is the 4th video. Okay, so I hope you get idea about the DNA replication. And if you like this video, make sure to hit the like button, share this video, and subscribe the channel to get more interesting videos like that. Thank you so much.